Hey everybody, thanks for joining. Stand by for just about 2 minutes. We're going to give enough time for folks to get into the room and we will get started. So thanks for joining. Hello everybody. We're just giving another 30 or so seconds. So attendees trickling in. We will get started shortly. All right, I think we're at a critical mass. Numbers are slowly dwindling in terms of showing up to the room. So without further ado, good afternoon, everybody. Thank you so much for joining. This is our 14th episode of When Things Go Wrong. I feel like every episode is special, but this one to me is especially special. And for that reason, because of the nature of the topic being with the amount of feedback and interest we've gotten, we did expand this to 90 minutes. So hang on, grab a drink, beverage of choice, snack, and bring your best energy to the room and attention. We are excited to launch a panel here today in just a moment. My name is David Valancourt. I run the GMP Collective and also serve as the vice chair for ASTM International's Committee D37 on Cannabis Standards. Today's topic is going to be focused on a particular ASTM standard or proposed ASTM standard that's going to ballot around what makes a cannabinoid potentially intoxicating. With that, I'm actually going to hand it over because we have an amazing special moderator today, Pam Epstein, who has been on our previous episodes. And I'm really excited and grateful to have her join. So Pam, why don't you come up and join, and while you get up there, before you kick it off, actually, a couple orders of housekeeping. Just wanted to share. Bring your questions. We got some great ones in the registration process, but there's the Q&A functionality in Zoom. So please bring your questions anonymous or shared. And as we have time and the conversation flows, Pam will be bringing them up and directing them towards our expert panelists. So Pam, over to you.
Thank you. Welcome, everyone. As David said, my name is Pam Epstein, and I'm the Chief Legal and Regulatory Officer for Terpene Belt. General disclaimer, I am not a scientist, and therefore these initial remarks are meant to set the stage and highlight the topic and prompt a robust and educational discussion with our esteemed panelists as we embark on a deeper understanding of why we need to have standardized treatment of cannabinoids irrespective of their source. In the realm of cannabis and hemp regulation, the discourse has recently reached a fever pitch. You can look on all the various social medias so that to know that we stand on the precipice of change. We stand on the next iteration of the Farm Bill. And these are active discussions. So within an overarching, I like to think of cannabinoid industry. We are all cannabinoid actors, and therefore it should not matter where you are sourcing your product from. We currently, though, find ourselves in a source-based house divided against itself, which echoes the words of one of my favorite presidents, Lincoln, who famously remarked that such a house cannot stand. It is precisely this division and the imperative to bridge it that underscores the paramount importance and the timeliness of today's webinar. It is with that I want to extend a heartfelt gratitude to the GMP Collective and especially the series when things go wrong, for bringing this distinguished panel of experts together and somehow allowing me to moderate them through this conversation. I'm joined today by leaders in the field of cannabinoid research, including the founder, as you've heard, and head of the GMP Collective, David Valancourt, and esteemed doctors Marcoux, Thomas, and El Soli. Collectively, they bring decades of experience and unparalleled expertise in unraveling the intricate science of cannabinoids during a time where cannabinoid research has been artificially constrained by the federal illegality. As they answer their first questions, I'm going to invite them to give a brief background on their robust experience. There is no better source of knowing where your information comes from than direct from the horse's mouth, as the saying goes. At the heart of the ongoing regulatory conundrum lies the elusive question of when a cannabinoid has the potential to induce intoxication, a critical factor that has contributed to the patchwork of regulations across the country and internationally. This is precisely the enigma that the ASTM standard, aptly entitled Metrics for Determining Intoxication and Non-Intoxication of Cannabinoids, seeks to address. For our discussion today, it is crucial to recognize that while laws and regulations are intended to safeguard the most vulnerable of our society, there is a parallel challenge. Just as there are individuals who do not consider themselves impaired, according to the blood alcohol content standard that we use, there is a determination of when a cannabinoid can be considered intoxicating is distinct from the assessment of impairment, which is typically a law enforcement metric, particularly in the context of the initial standard that we are exploring today. Throughout this webinar, we will delve deep into the nuances of terminology and nomenclature with our panel of experts. In addition to exploring other scope exclusions, vital to establishing this is a foundational standard. It is one of many. This webinar represents the significant and critical step forward. We will have a longer and chain of standards that will shape the future of cannabinoid regulation, driven by its guiding principle of science and safety. Before we commence, I'm going to reiterate some housekeeping issues from David. If you have any questions during the presentation, please make sure to put them in the Q&A and I will incorporate them to the best of my ability. And with that, I'm hoping you can all join me in extending a warm welcome to the esteemed panelists we have as we embark on this scientifically driven exploration of the principles that are underpinning the standard that will safeguard public health and safety in the realm of all cannabinoids. So to start, I'm going to throw it back over to David, and I'm going to ask him to provide us with an overview of the standard specification and explain why it is essential for the cannabinoid industry, and to highlight for us some of the reasons that I've initially discussed as to the absence, what has created the chaos in the absence from such a standard.
Well, thank you so much, Pam, for that great introduction and passing it back to me. So, you know, the previous episode we had, which is available on our YouTube channel, we talked about the ASTM process. So recognizing this is a voluntary consensus standard being proposed, and with the specific scope being around how to define how to classify whether a cannabinoid, regardless of its source, has the potential to be intoxicating. And I think whether you're in the United States or internationally, we've seen that a lot has evolved since Delta-9, the days of Delta-9 and CBD being the two primary cannabinoids of interest, both from a consumer interest and being available in the marketplace. We have, Delta-8, Delta-10. We have other isomers of the tetrahydrocannabinols. We have minor cannabinoids that exist in the plant in various quantities, and the potential for intoxication, as we'll dive into what that means. Without that basic foundation of how to determine whether a cannabinoid may potentially be intoxicating, we can't go on to a second step of diving into, things like source of route of administration, things like, the quantity of it or concentration of it in a product, to be able to determine public health and safety. Because really what this is all about is consumer trust and transparency and public health and safety. And when we have a basic set of guidelines on how to determine this criteria, And then we can all work together with that framework more efficiently and effectively, whether you're in a regulator's seat or industry or even a consumer, recognizing how this might have implications for warning symbols or age gating or otherwise. And that's, I think, especially prevalent as we've seen in the hemp-derived marketplace, where potentially intoxicating products are being placed in gas stations and other locations without any regulatory oversight. And we're not here to say this is bad, this is a voice of no, but this is how to put some guardrails around and classify things so we can move forward as an industry. So I hope that's a good kind of overview context. I'll back over to you, Pam.
Yeah, I always think it's important, in order to say what isn't, you have to know what is. So we're sort of not letting the enemy be the perfect, or not letting perfect be the enemy of the good. And we just, to your point, really need some guardrails there, because as a consumer, I think it is fair to say that most people would believe that our federal government wouldn't allow products into the marketplace that would get you high, right? That's what that 0.3% is supposed to be de minimis. So with that, I want to turn to Dr. Marcoux, and I'm going to ask him to not forget to give a slight introduction. And now that we have sort of a baseline understanding of the standard, I really want to dive into some of the exclusions and the terminology that we're using to distinguish between impairment and intoxication, how they relate to each other, and how they are going to be utilized in the standard. Because I know there's a lot of kind of questions about why we've chosen one word over another, and you may want to throw in psychoactive. I'm going to leave that up to you.
Well, thank you. So yeah, my name is Jayon Marcoux. My PhD is in molecular pharmacology. I study the structure and function of cannabinoid receptors, and actually even Mahmoud Al-Soli, while I was in grad school, actually allowed me to characterize some compounds to help him out with a project. So I'm very familiar with screening and characterizing compounds, as is Dr. Brian Thomas as well, who you'll hear from later. I am a basic and clinical researcher. I'm also a court-qualified expert witness. One of the fun things about being a court-qualified expert witness is you get cited in case law. And I'm actually cited in case law about how changes to cannabinoids affect their activity. Even small changes affect their regulatory status and their activity. So I'm going to start by talking about what's not in the standard. We'll go through some definitions of impairments. I've tried to prepare some fun analogies to disentangle this sort of context-dependent terminology, and then go into just ending with why this standard and why now. So again, the intention of the standard is to provide a foundation for future work in this area, helping, as David pointed out, consumers understand what is the amount that someone might get impaired or intoxicated about. And I think I'm going to stick with intoxicated and define that later. But again, this is to help and provide metrics that can be utilized to promote consumer awareness and protect public health and safety through a sort of total intoxicating cannabinoid concentration, or TIC number. I like to think of it, TIC, like it's sucking the sobriety out of you. Just a little bit of humor, right? We can laugh, it's okay. The standard currently focuses only on the inherent properties of cannabinoids. It does not consider variables right now, such as route of administration, serving size, or potential tiers between these different cannabinoids. So these are factors we want to include and should be addressed, assuming that these intoxicating properties are, can be proved otherwise. And again, where does this standard come from? It comes from scientific experts, academic experts, industry experts, and regulators. Again, this standard is to serve as a reference for compliance, labeling, and largely consumer awareness. So impairment and intoxication, what is the difference, right? So when we think about intoxication, the simplest definition is that intoxication is basically being defined as being under the influence of drugs or alcohol, while impairment is more about your mental or physical abilities being affected. So intoxication can lead to impairment, but impairment doesn't always lead to intoxication. I'll repeat that. Intoxication can lead to impairment, but impairment doesn't always lead to intoxication. I will use a few fun analogies I've been scrubbing the internet for, and here here's one that I think perhaps will resonate with people. Impairment. If for those of you who play guitar or musical instruments, if you're picking the strings for 8 hours, your fingers will get tired. Your ability to play efficiently will become impaired. If you put a glove on your hand and then try to play those chords, your hand will be more intoxicated, right? That will affect the performance much more significantly. Use a fuel gauge analogy, right? Impairment is like driving your car in a low fuel gauge. The physical abilities of the car might be running on low, making it challenging to function efficiently. On the other hand, intoxication would be putting the wrong type of fuel in your car, and the engine behaves erratically and unpredictability. Can't talk today. Must be getting impaired from talking about this all day. So, similar with computer analogies, right? If you have 50 tabs open, your computer's ability to process those tabs will be impaired. However, if you download a virus or malware, your computer will become intoxicated, not functioning as predicted, erratic behavior and whatnot. The analogies go on, For those of you who still have bunny ears on your television, here's one for you. Impairment is like having a slightly fuzzy TV signal. You can still watch your favorite shows, but intoxication, would be like losing the whole TV signal in its entirety. And again, you can come up with your own. We could talk about cell phone batteries, right? Low cell phone battery. Some functions get impaired. It goes into low power mode. But should something drastic happen to your phone and lose all functionality, that would be more like intoxication. So I hope, these analogies are meant to highlight the varying degrees of impairment and intoxication. And since impairment is more of a natural thing that just happens, I wake up in the morning before my first cup of coffee, I would say I am impaired, but I'm not intoxicated. See, that's more of like being sleepy, you can be impaired. That's not against the law to be sleepy in most states yet. So, but I think that's the difference. So we can't talk about intoxication because that is literally in reference to a drug or something acting upon you. So why create this standard? I think as the industry grows, it's becoming stranger, and these patterns are becoming more complicated. Claims about safety, efficacy, and even the nature of hemp products and derived isomers soared so far above the data. It's really crazy, and I think we're experiencing a bit of a terminology context collapse in this space around how we talk about it. And I guess, why am I working on this standard? I think it's also important because I don't know, we vote on these. It's a consensus process, and everyone kind of has their own reasons. And, in graduate school, I was relieved to find molecular pharmacology. It was a concrete enterprise, cause and effect, put a drug on a receptor, super easy, right? I don't feel like we have that now with the way products are labeled, discussed, and even the nature of the compounds that are in a lot of products. But one of the reasons I think this is important is me and my colleagues did some research, which we shared at a conference, and we looked at adverse events reporting in national systems and poison control centers for substances. And one hemp isomer in just 2 or 3 years had more adverse events reported by consumers than psilocybin had in 40. So let's think about that. Magic mushrooms, been around for a while, order of magnitude higher in just 2 or 3 years of adverse events reported for a single hemp isomer product. That is a safety signal I don't think we can ignore. And maybe it comes down to consumer education. So why have the standard? Why now? Last analogy, I promise. and I'll just use one last colorful analogy because I love them so much. And I like to think of the mythological creature, the satyr, coming out of the woodwork, right? Coming out of the woods with some earthly delights in its hand. And we're sitting there saying, is that wine or is it grapes? It would be really good to know because some of us want grapes, others want wine. Some of us would like to get grapes and not have it be wine, and vice versa. And I think that is really why we're here. People want to know what they're getting, and I think that is the essential thing. So I'm gonna pause here and pass the mic back.
That was perfect. I really appreciate making the real world connections with analogies. I'm a big fan of comparisons as well. For all of you in the room, there's a lot of alphabet soup that happens in every industry. So the TIC number is a total intoxicating cannabinoid concentration. which we hope that will eventually be adopted. I love the idea of terminology paralysis, because that's really sort of what is happening. And with the ubiquitousness within consumers, that's really what we're looking for, is to educate them. I think what Dr. Marcoux was mentioning is the National Centers for Poison Control issued a new product code in 2021 for Delta 8. They don't undertake that process lightly, and that means that they had so many calls that they had to specifically identify it. But within understanding, now that we have some of that science moving into a scientific perspective, we understand the why and the how intoxication and impairment work. I want to explore the historical content, and I want to call on Dr. Thomas, who will introduce himself at the same time, to discuss the scientific understanding of intoxicating effects of cannabinoids and how they've evolved over time, highlighting key milestones, including the introduction of one of the foundations of the standard, the tetrad assay, and the discovery of our endocannabinoid system and the CB1 and CB2 receptors.
Thank you, Pam, and greetings to everyone. My name is Brian Thomas. I have a PhD in pharmacology and toxicology. I got that from the Medical College of Virginia, Virginia Commonwealth University, and my dissertation research involved studying the mechanism of action of cannabinoids. My employment background includes almost 30 years of service as a research scientist and senior director of analytical chemistry and pharmaceutics at Research Triangle Institute, which is a not-for-profit contract research organization in North Carolina, where I am today. And I continued while at Research Triangle Institute, our RTI, I continued to be involved in cannabis and cannabinoid research funded by the National Institute on Drug Abuse, the Drug Enforcement Administration, and a lot of other grant and contract research sponsors. I have also over 5 years of more recent experience working in the Canadian cannabis industry. And I'm currently providing consulting services to the industry through Empirical Pharmaceutical Services. And I serve on the scientific advisory board of NALU Bio and a couple of other pharmaceutical cannabis concerns. I have published over 120 peer-reviewed research articles, the majority of which have been focused on cannabinoid receptor pharmacology. And it is my pleasure to be here today with the distinguished colleagues and with the audience here. I do have a lot of history in cannabinoid research, so I think it's probably apt that I can describe a little bit of the history in the characterization and definition of cannabinoids. And we all know that cannabis and its various formulations have been used for thousands of years for recreational or medicinal purposes. But it really wasn't until the early 20th century that the chemical constituents of cannabis sativa began to be purified. Their structure and pharmacological activity systematically elucidated. Importantly, the structure of cannabinol had been accurately determined, and a wide variety of extremely potent synthetic cannabinoid analogs had been synthesized and could produce profound effects, shown to produce profound effects similar to those observed with cannabis in laboratory animals and in man. It wasn't until the 1960s when Rafi Meshulam ultimately elucidated the structure of minus trans delta-9 THC, one of the 4 possible stereoisomers of delta-9 THC, and he identified it as one of the primary active ingredients in cannabis that could replicate its unique spectrum of pharmacological effects, which includes changes in perception or the high, and drowsiness and central nervous system depression, analgesia. We're familiar with increased appetite, anti-emesis, anti-vomiting, and also detriments in cognition, memory, and psychomotor performance. changes in blood pressure, intraocular pressure, heart rate, respiration, and body temperature are also typically observed, along with red eyes, dilated pupils, and drooping eyelids. In laboratory animals, Pam had mentioned, wanted to know some of the key milestones. Laboratory animals, it was quickly recognized that the changes induced by minus trans delta 9 THC included profound static ataxia in canines. And it also produced catalepsy, analgesia, hypothermia, and altered locomotor activity in rodents. And that's the catalepsy, or lack of movement, analgesia, hypothermia, and decreases in spontaneous locomotor activity represents something that my mentor from graduate school called the tetrad of cannabinoid effects. I had mentioned in the 40s that some extremely potent synthetic analogs of cannabinoids were made by Roger Adams and his colleagues. And back then, people took them and reported anecdotally the effects that they observed when they went home. So they're recorded, and one can read those. To tell you about the type of potency that they were able to achieve in 1940, there was a dimethylheptyl delta 6A10A THC isomer that they made that with a 10 milligram per kilogram IV administration in canines, it would put them unconscious and reduce their body temperature for 6 days, and they would recover uneventfully. So laboratory animals have long been used to characterize the effects. And while these in vivo and in vitro effects of cannabinoids were becoming increasingly well described through the mid 20th century, it wasn't until the late 1980s that Bill Devane and Lynn Howlett discovered the CB1 receptor that is densely distributed in your brain and your peripheral nervous system. And Bill and Lynn established that this G protein coupled receptor is one of the principal mechanisms of action of minus trans delta 9 THC. And specifically, they and others showed that the ability of compounds to bind to and activate the CB1 receptor, which we consider that their affinity and efficacy as agonists. Their ability to bind to is their affinity, and their ability to activate the CB1 receptor is their ability, their efficacy as agonists. Those two properties, affinity and efficacy, were shown to be correlated to their ability to produce behavioral effects in animal models, as well as correlated to their psychoactive potency in humans. And this was demonstrated over and over again with a wide range of phytocannabinoids and synthetic cannabinoid analogs. In the 1990s, the second cannabinoid receptor, CB2, was identified. It's not densely expressed in the brain, but it's in the immune cells and macrophages in the spleen. And this accounts for some of the non-centrally mediated immunomodulatory actions of cannabinoids. During the same time period, though, Bill Devane was overworking with Rafi Meshulam, and they identified endogenous cannabinoids in the brain that also bound to and activated the CB1 receptor. And again, it was shown that the IV administration of these endogenous compounds could replicate the in vivo effects observed with minus trans delta 9 THC. So you start to see the theme. If it can bind to the CB1 receptor, if it can activate it, whether it's exogenous, whether it's endogenous, whether it's synthetically produced, whether it's manufactured in the plant, these compounds have the capacity to produce the pharmacological effects that are observed with minus trans delta 9 THC. And then finally, also in the 1900s, cannabinoid receptor antagonists and inverse agonists were discovered. And just like naloxone is to fentanyl overdose, cannabinoid receptor antagonists can block the effects of minus trans delta 9 THC. And CB1 receptor inverse agonists, and their name implies what they'll do, inverse agonists, they can produce pharmacological effects opposite to those observed with minus trans delta 9 THC and other cannabinoid receptor agonists. So the one cannabinoid receptor antagonist made it to the market and was sold in Europe to help people lose weight and to help with metabolic syndrome and the associated aspects of diabetes. But unfortunately, it also produced the opposite of euphoria and the high. It tended to make people depressed. And so it had to be withdrawn from the market. It was not approved by the FDA for the market in the United States. But as a result of all of this research, it is widely recognized and appreciated that compounds that bind to and activate the CB1 receptor can produce a relatively unique spectrum of pharmacological effects in vivo, the tetrad effects, psychoactivity in man, and that these effects can be selectively blocked by CB1 receptor antagonists or inverse agonists. And it was very clear when one could find an effect, a pharmacological effect in an animal and block it with a cannabinoid receptor antagonist, that was a clear indication that it was CB1 receptor mediated or CB2 receptor mediated, depending upon the nature of the antagonist. So I hope this helps give a little historical perspective as to how these things kind of fell in line over time and how the evidence just became increasingly compelling that this CB1 receptor that is extremely densely distributed throughout your brain and mine is the biological substrate that delta 9 THC interacts with and produces its pharmacological effects. So Pam, I'll hand it back to you.
Dr. Thomas, I need a glossary of terms, but I think that everybody always is getting educated around the history of cannabinoids. I think we all interact with them every day, and we don't really understand how they've come to the forefront. So I really appreciate you walking us through that history. I know for myself, I learned a lot in those last 5 minutes. And I'm going to take that as a practical application and ask Dr. Al-Soli to, while considering that practical side, to share some insights on how the standard can be implemented in real world scenarios. What implications might it have for product labeling and consumer safety, specifically referencing the paper you recently had published, Isolation and Characterization of Impurities in Commercially Marketed Delta 8 THC Products, which was in the National Products Journal, and I read and found completely fascinating. I'm sure we can get people access to that, but I'd love to have your thoughts. And a brief introduction, don't forget.
Thank you very much, Pam, and thanks for everybody. I enjoyed Dr. Marco and Dr. Thomas, presentations and elaboration on the history and the pharmacology of the psychoactive cannabinoids. My name is Mahmoud Al-Soli. I am the director of the marijuana project at the University of Mississippi, which is a contract with the National Institute on Drug Abuse to provide standardized materials for research, whether it's plant material, extracts, pure cannabinoids. We do all this for the drug supply program at the National Institute on Drug Abuse. And I enjoyed the time that Dr. Thomas was working at RTI. We collaborated quite a bit. We talked a lot. We visited quite a bit. And so I appreciate his presentation. And of course, Dr. Marco, I've known him for years when he was in graduate school, and he did look at some of the cannabinoids that I produce in my lab. So we all have very much a connection that goes back many years. I am a research professor at the National Center for Natural Products Research at the School of Pharmacy, University of Mississippi, and I'm also professor of pharmaceutics and drug delivery at the same school. My background actually is in natural products chemistry. I graduated from the University of Pittsburgh back in 1975 with a PhD in pharmacognosy, not pharmacology. People don't really quite make the difference between pharmacology and pharmacognosy. While pharmacology is the dealing with the effects of drugs on the different parts of the human body, pharmacognosy is more or less like a PhD in natural products chemistry. And so that is my background. I moved to University of Mississippi back in 1975, and I worked on so many different areas. But joined the marijuana research back in 1976 and participated in the isolation and structure elucidation of the majority of the known cannabinoids today. While delta-9 THC and CBD, as Dr. Marco indicated, these are the two most studied compounds. The cannabis plant actually contains over 120 or 127 today different cannabinoids, and we only know the pharmacology of a few of those cannabinoids. Fortunately or unfortunately, there is only 5 or 6 major cannabinoids, and the other ones are minor cannabinoids. But how much contribution these other cannabinoids have on the overall pharmacology of the plant is really not very well known. But it's fair to say that the major pharmacology of the cannabis plant that is on the market today is expressed by the amount of delta-9 THC that exists in the plant material. I have over 400 publications in the scientific literature. I have over 100 patents in this area, also looking at developing different drugs, cannabis based or originated in drugs and pharmaceuticals. And so I have quite a bit of work in this area. I haven't done a lot of pharmacology work, but I have been associated with people that have done a lot of that, and therefore their knowledge rubs off a little bit on me. So I'm going to take this topic following Dr. Marcos and Dr. Thomas's presentations and say that the effect of intoxication or impairment, whichever one you want to look at it, of course, there is, as they both indicated, the parameters that could be used to show or indicate those types of activities. But those activities, I want to add to what they said already, is really dependent on, and Dr. Marco referred to that as the dose of the material that is actually being consumed. That is absolutely true, but we also have to consider 2 other factors that come to play in getting the effects of the drug, whether it's THC, natural cannabinoid, or synthetic cannabinoids, and that is we talked a little bit about the route of administration. Of course, if you smoke the material, then you get the drug very quickly into your system, and therefore the impairment or the intoxication happens a lot quicker. If you take it orally, then there is a lag period between the time the person consumes the drug to the time that actually the highest blood levels and brain levels of the drug are achieved that to produce the intoxication or the impairment. But the other thing that we really need to be thinking about, not only the dose and the route of administration, but we also need to think about the tolerance that the people have. While somebody can consume 3 or 4 shots of whiskey and still be okay, I need a little bit more to reach the level of intoxication that they need to get. While somebody like me, I can drink a non-alcoholic beer with 0.5% alcohol, and I can feel the effect of that 0.5%. So the tolerance is a very important factor in determining the true impairment. Yes, the person is under the effects of the drug, but whether they are really reach the intoxication level or not is a function of the person's tolerance or ability to tolerate the drug. And of course, this is very common both in the alcohol area and in the cannabis area. The other thing is that we need to be thinking about in determining intoxication and impairment is the person's own ability to metabolize the drug. The variability between the inter-subject variability is huge, both in the animal models and in the human model. That while some person can be able to metabolize the drug very quickly and get it out, another person doesn't, so it stays longer in the system. So the metabolism, the inter-subject variability is another important factor in determining intoxication and impairment.
I think
go ahead.
Go ahead. No, I did not mean to interrupt you. Continue.
Okay, no problem. Okay, now we have, as I mentioned, we have a lot of cannabinoids that we don't really know the actual pharmacological activity and their ability to cause intoxication or impairment. But we know you mentioned and Dr. Marco mentioned on the delta-8, which is the problem that is caused right now, and the overdoses and the side effects and so on. Delta-8 THC on the market, and it's referred to as a hemp product. It really is, between the 5 of us or 6 of us, it really not a hemp derived product. It's a compound that is derived from a hemp derived product. They take the CBD, which is a hemp derived product, hemp-derived cannabinoid, and they convert that CBD into the delta-8 by chemical conversions. It doesn't mean that delta-8 is actually present in any level, that's really measurable in the hemp plant material. It might exist a little bit in the regular cannabis or marijuana, but not to the extent that you can actually isolate enough material there to put on the market as a product. So all the delta-8 that exists on the market today is either totally synthetic or derived from CBD conversion of CBD to delta-8 THC. We were approached by the USP to look into this issue of contamination into the delta-8 products on the market, and we did find out that the products that are marketed as delta-8 products, they have wide range of different impurities in there, and we were able to isolate maybe, I think it total is maybe 12 different impurities out of that the commercial delta-8. And we actually have a grant proposal that we just submitted to NIH to look at the pharmacology of those different components that exist in the delta-8 in terms of their pharmacological activity, their intoxicating ability, if you will, and also their interaction with delta-8. What does each one of those components do to the activity of delta-8? So these are some of the things that we are working on right now.
There was a good question that came up in the chat that I want to ask, and one that I think everybody is always interested in is delta-8 a naturally occurring cannabinoid? So I'm going to throw out my understanding, and I'm going to hope that you guys are going to either confirm or correct me. So as I understand it, delta-8 can occur naturally in the cannabis plant. It can be a degradation as the plant goes through its normal synthesis, but it does not occur in commercially viable quantities. So the prevalence of delta-8 on the marketplace is generally a converted cannabinoid as like a lay consumer would understand it, or as I would understand it. So I'm going to throw it out to the group to kind of validate, or feel free to correct me, because I'd love to be able to say that accurately in conversations, because it comes up often and the courts have grappled with, is this a naturally occurring cannabinoid or not?
I think if there was a plant that naturally produced high amounts of Delta-8, it would either be growing at the University of Mississippi or it had been discovered 40 years ago somewhere else. These things have been around for a while. GW Pharmaceuticals probably would have published about it. I mean, they published, ATN de Myers published so much about the genetics of CBG only plants. The propyl analog varieties, and even cannabinoid-free plants. So we have no evidence that really Delta-8 occurs in significant amounts. I mean, maybe if you grew 20 tons of hemp, you might be able to extract enough Delta-8 for like one product run, maybe. I don't know. I'm just back envelope calculation there.
And I think, Mahmoud, you can correct me if I'm wrong, but there is some isomerization that occurs to Delta-8 THC over time, but it's an extremely slow process and produces only a very small amount. Again, to Jahan's point, you'd have to grow a lot of cannabis in order to then take all the effort to purify the very small amounts of Delta-8 THC. Am I incorrect, Mahmoud?
No, you are absolutely correct, and that's really what I indicated in my introduction here, is that the Delta-8 that exists in the plant material is really not naturally produced by the plant. The plant produces Delta-9 THC, which, because the double bond between the 8 and the 9 position, between the 9 and the 10 position, which makes it Delta-9 THC, that product in the isomers of the tetrahydrocannabinols is not the thermodynamically stable isomer. So it's a first one that gets formed, but then if you heat that material or you expose it to acid, it's going to move to the Delta-8 position between the 8 and the 9 position, that's the thermodynamically more stable isomer. So the natural plant, whether it's marijuana or hemp, produce Delta-9 THC. Of course, marijuana produces a lot more, but hemp does have some level of Delta-9 THC that might, a little bit of it might isomerize to Delta-8. So it's very well known from day one, from the time that I joined and started working with cannabis, that Delta-8 THC is an artifact that is produced in very, very small amounts by conversion from Delta-9. But if you take Delta-9, as you very well know, Brian, and you put Delta-9 in, let's say, methylene chloride, and you add a little acid to that, you're going to get mostly converted to Delta-8. So Delta-8, because the double bond moves very quickly, in the presence of acid to the Delta-8 side, and therefore that's how you form Delta-8. But to answer your point, Pam, Delta-8 THC does not exist in any significant amount in marijuana, which has a high level of tetrahydrocannabinol, Delta-9, let alone in hemp. I mean, hemp has a very small amount of Delta-9 THC to start with, and even a much more minute amount will be formed in hemp. So this idea that's coming from hemp as a natural, you just crash that totally.
This is where the courts could receive some expert witness testimony. Dr. Marco, I'm looking at you. They need this kind of information to understand the difference between what is naturally occurring and what is commercially viable. If we look at comparable industries like dietary supplement law and geraniol oil with ephedra did not allow you to usurp safety data when there was not cultivar over cultivar commercial viability. So which leads me to a question about public health and safety. As we're talking about intoxication and how all of these things relate, the difference between phytocannabinoids and converted cannabinoids, and leads to a question that we had in the Q&A, which is there a utility in distinguishing between semi-synthetic and synthetic compounds? And I would actually broaden that out to segmentation into buckets of phytocannabinoids that are naturally occurring and commercially viable, separate and distinct from those that may be naturally occurring but not in commercial viability, and then those that are wholly like synthetic that don't occur in the plant at all, let's say like THCP, and whether or not from your academic research, scientific driven data set, how we should be approaching these from a safety perspective. And I'm going to throw that out to the group, whoever wants to take it on.
I'll take a first swing at that, and definitely want to leave room for Dr. Thomas and Dr. El-Soli to talk. But if I was to think about these compounds, the reason why the distinctions are important, and this almost gets back to kind of what actually Dave Valancourt does, is it gets back to the quality of the product. What's in the product? If we know it's a semi-synthetic product, we know there'll probably be footprints, artifacts from that synthesis. We've seen that with some of the products out there. A lot of olivitol, a lot of other byproducts, a lot of accidental THC made as well, delta-9 THC, just to clarify, when people are trying to make other THC compounds. So I think those distinctions are important for the quality tests, for the quality control, for the specifications of that product. So if we know that there is something made during the processing of it that needs to be tested for that shouldn't be there, that those classifications become important for those little things we call certificates of analysis.
It was really well said. I agree completely. They're like industry best practices for each of those buckets and expectations, and the consumer shouldn't necessarily be in the position to understand all the nuances and risks associated with where the product came from, how it was isolated, and instead, I think we need to put the onus on the industry to own up, and state things correctly, and then follow the expectations. Like Jehan had said, if you manufacture something synthetically, it's just good standard practice to identify impurities above a certain level and to monitor the level of impurities, even if you don't identify them, if they fall below a certain amount. So there's a lot of good manufacturing practices that are applicable for cannabis, agriculture, for cannabinoid extraction and purification, for synthesis, et cetera. So I think it's really essential that we all recognize that these compounds may not be, hemp derived. It's so easily said, but again, as Mahmoud said, they're derived from something that was, isolated from hemp, not the same thing as hemp derived.
It's the phytocannabinoid versus the downstream derivative and then conversion of a conversion. I think what you guys are all saying is truth and honesty, right? Truth in advertising, validation, and the reason that ASTM, the 125-year-old organization that it is, imposes standards, right? Wants to bring to the forefront and lead by science. I don't think anybody wants to vilify a cannabinoid. I don't know what any of these amazing cannabinoids have the potential to do. But the closer we get to a pharmacological drug, the more that we should be aligning ourselves with that level of safety, right? When you take a Vicodin, you know that it is 99.9% of the Vicodin, and if it's Vicodin / hydrocodone or acetaminophen, you know what those ratios are. Nobody's guessing. at this game. And that's currently what we have devoid in the marketplace. I know David says it to me all the time. It's not about the poison, it's about the dose. And so I want to open up to everybody. I know
just
that
add briefly one thing.
David,
Yeah.
go ahead. Yes.
Thanks. So yeah, just on the semi-synthetic versus, phytocannabinoid and, source of cannabinoid, I think there's two things to keep in mind. One, if we go back to the work item or the proposed standard around a potentially intoxicating cannabinoid, regardless of whether it's a semi-synthetic or synthetically derived or whatever term we agree on, it does not make a difference in terms of its potential for intoxication. With a little asterisk that I know I had a great conversation with Brian more recently, reminding us that there's 4 flavor stereoisomers, right, of delta-9 THC. And, if we want to get down to that granular of a level, there might be different, response factors to, put it simply, between those. But for all intents and purposes, let's talk about single, delta-9, delta-8, CBD. They're going to have the same yes, no, potentially intoxicating. Now, why it matters, though, still to consider it is the other context of what's in these products and what should we be looking for. So it's back to those batch records and how you're producing. So if you're producing a semi-synthetic cannabinoid, that means there's other solvents that are being used that we need to know to look for to ensure that there's public health and safety that would not necessarily need to be there, need to be looked for if it's a phytocannabinoid, 100%, phytocannabinoid product where no other solvents, acids, or otherwise were used. And I think, if you even think of an industry standpoint, let's not test for things that there is a 0% chance that it could be in there. Let's look for impurities that may potentially be there, which is why we need to understand those production processes. Brian, I saw you come off of mute, so go ahead. If you want to.
Only just because I tried during my discussion earlier to constantly say minus trans delta-9 THC, because THC that's a lot of compounds, right? And then with delta-9 THC, there are 4 possible isomers of delta-9 THC. So when you talk about 4 and only 1 has significant pharmacological activity. So it's just if we use the terms loosely, as long as everybody knows that you're talking about the one, that's okay. But when you say THC, and in real chemistry, there's a lot, and they all have the potential to have widely different pharmacological activity.
I fully agree with you, Brian, on that. I think it's just the matter, the common, let's say the common knowledge, if you will, among the ones working in the area of cannabis and cannabinoids. When you say THC, you mean minus trans delta-9 THC. I mean, that's the, when you just say THC and you don't have any other acronym with it, it's the one that you are referring to, the minus delta-9 trans THC. And so and only one of the transes, because you can have 2 transes opposite to each other, but the one that exists in the plant material is the one that just about everybody refers to as THC. When you say just THC, at least in my own mind, I'm thinking of that one particular one that you're referring to.
David, I'm thinking we could have a webinar on each one of these topics separately, but what's highlighted through this discussion is really to remember that this is an initial standard just to determine if there is the potential for this cannabinoid to be intoxicating, and downstream we need standards across the board around manufacturing, around heightened impurities, whether they're going to exist from a plant material, a phytocannabinoid, versus something that is manufactured or further chemically produced. So with that, I'm going to open up a question to ask, since we have this inconsistent, I would say, treatment in between sources, right? In the cannabis space, we're burdened with all of these quality controls. There's QA/QC testing in order to pass the gate to get to the consumer. And in hemp, we're really looking towards the industry to establish standards and not have a race to the bottom for profit, because there is no gating factor between those products and the consumer, right? They go right from the manufacturer direct to the consumer without having to pass through a quality assurance gate. So how can the scientific community and the industry come together to collaborate and ensure public health? And I'm going to ask that maybe you all opine on that, because you're coming at it from a different perspective, and really important for the regulators and the industry in the room to understand how this nexus, this inflection point comes together, because everybody's trying to do right, especially the regulators, by the consumer.
I'll tee it off again, and then I'll let the other legends on this webinar follow up. Let me know if I got it right. So I think how I'd start by answering that question is that this is a question for this generation and the next generation of people in this industry. The people are going to be the experts, the people are the experts, the operators, the academics. And the question is not whether or not our system is perfect for regulating and understanding these products. It's what to do about those imperfections. This standard is intended to find an alignment between pharmacology and politics. And I think for those of us who believe cannabinoids are more than an exuberance-themed consumer product, and I think it's always good to remember that the issues we are facing come from a messed up regulatory and political history. And it's this history, the system that has coughed up these compounds we've been talking about today, these products of interest and concern, among other issues. And I think the future really is we have to embrace the standards development process and the scientific process to be able to see the challenges that are going to be here for the next decade or so. And I think also, probably the strongest allies, I think, for academics and for operators who want to be compliant with regulations and pursue best practices are regulators and are those state regulators who in some ways have been getting, who have signed on for a career to defy federal law to run these state programs and to make sure those businesses succeed. They succeed because when the industry succeeds. So I think that's also important to look at for success in addressing some of these issues. We have this golden age of science, but yet we can't reliably predict what will happen when someone consumes a hemp product. Some people are fine, some people get sick, some people get intoxicated more than others, and we don't know why. And I think that we would really like to know why. And I think it's working together with the researchers, the industry, and the regulators.
May I add something to what you said, Johan? I think the industry really bears a very, very big responsibility on this. A few years ago, I think maybe 4 or 5 years ago, I embarked on looking at hemp-related products or whatever in the cannabis industry to analyze it and see what's in there. Does the amount that they put on the label is truly what's in there in the product? And make a long story short, along the line of doing this, I saw some products that are labeled hemp products. And I look at them, like CBD related products. And I looked at them and I don't see no CBD. I don't see even no THC, but I saw some strange peaks that I don't normally see in this product. And I take this and go through the extra mile just to find out from my scientific curiosity, if you will, to say what's that. And this ended up to be actually synthetic cannabinoid. So here we have a product that's supposed to be a hemp product, supposed to be a CBD product, and end up to be a synthetic cannabinoid product. So that's that's not even something that maybe doesn't have good quality control in it. That's just plainly cheating, deceiving the public with putting some materials in a product that's totally unrelated. It doesn't say anything about hemp. It doesn't belong anything where in the hemp. It's something that we put there to intoxicate the person who is ingesting this product, getting him to be hooked on that product, and come back over and over and over to buy the same product because they're getting high on this product. So the industry, I think, has a very big responsibility, and it's really difficult. There is thousands and thousands of cannabis industries in this country that it's very difficult to regulate. So unless the industry itself takes responsibility and do the right thing, you might be able to catch some industries that are not doing right, but you're not going to be able to catch all of them. It's very difficult. So the industry really needs to be more responsible in putting products on the market that are what they say they are, at least. So that's my position.
Mahmoud, I agree so much with that. And, I think of these stories about the bus driver who thought he was eating a non-intoxicating product. I mean, it doesn't matter until it happens in your community, right? The truck drivers who are driving big trucks and picking up something at a truck stop thinking it's non-intoxicating. And then they either fail that drug test down the road or are driving unsafely. And these are issues. You don't have to believe me. You can look it up on the internet. Why trust the experts when you can ask Dr. Google or whatever? And these are not issues. I mean, I've received calls from people looking for experts to help them because they're in a legal bind. People get pulled over and they're, unbeknownst to them, there's THC in a product in a state that doesn't allow THC or other compounds, as you point out, in the product, and they go to jail, sometimes for weeks at a time. They lose their job, they lose their livelihood, all because a product wasn't sufficiently labeled or they weren't sufficiently educated about the product. It can be a dangerous buyer beware depending where you live. And so I think that is one of the great things that this standard might bring is clarity to that. And people aren't going to be playing this sort of, drug of the month club when they use these products. Like, what's in it? Is it going to be safe for my grandmother to use or not? Like, those are questions I think that are important. Is this something we want truck drivers to be able to just eat by the handful because they're available at gas stations? These are important questions, and having education. And the last thing I guess I'd say about this is pharma. Pharma is required to educate about their drugs. We all feel our own feelings about pharma, but if they put out a product that was never tested in humans, they did not provide education to clinicians, we'd be really, really upset. But yet we accept it for some drugs and not others. It reminds me of something I've seen Dr. Amanda Ryman talk about, which is drug exceptionalism, where some people believe that their drug or their product should be the only legal one, should have no restrictions, and this is how it should be, but everything else should follow a different set of rules. And we're just looking for consistency in how we approach this.
This is where the standard will become pivotal. It becomes the scientific adult in the room. Unfortunately, what has happened is the 2018 Farm Bill gave a very open-ended allowance for hemp. And that allowance was for seed, stalk, fiber, and non-psychoactive CBD. It has become a Trojan horse for intoxicants. And as we started at the top of the call, we are all a cannabinoid industry. There is no reason that these products cannot exist, but they need to exist in sensible regulation. And that's what we need to support the regulators in being able to see the forest from the trees, because Dr. Marcoux, you gave a great example of truck drivers. This is also about children, right? And unintended consequences. When a product is labeled hemp, and we've seen some in the hemp community suggesting, and I'd like to hear the experts' thoughts on this, that 5 milligrams of THC Delta 9 is non-intoxicating. There's a current prevailing white paper that has been issued by one of the leading hemp trade organizations that suggests that 5 milligrams is a non-intoxicating dose of a product in a serving size. So I'd like to get your thoughts on that from a very scientific aspect, and say that this is what the standard is going to try to evaluate as a baseline. where that threshold of intoxication should suggest, and the moral imperative, right? We're entering a moral imperative. Just because you are arguably, by the ambit of language, allowed to do something, does that mean that you should do it, or that as a country, we do not have the awareness to change and evolve as the science leads us there? So I'm going to open end that to have you guys help opine on that particular request of an industry.
I mean, if 5 milligrams did not cause intoxication, I don't think, I think Marinol would be in a less restrictive category than Schedule 3. Why would it be in the package insert for 5 milligrams of Marinol, which is a form of THC? But maybe Brian, yeah.
Well, I think once again, Jehan's right, and certainly their 5 milligrams has the capacity to be intoxicating. If you rub it on your arm, and it's probably not going to be, right? So, our standard is with respect to can things be classified as potentially intoxicating at this point in time? Again, the unit doses, I suspect that, one might consider, 5 milligrams to be a unit dose that might be associated with intoxication if one consumed it. But again, that, one might have to, understand that there'd be a lot of variability if you consumed it on an empty stomach, if you weighed 20 kilograms compared to 70 kilograms. There's all sorts of factors, if you're male or if you're female, that would determine one's sensitivity to a 5 milligram dose. And once again, I think, to your point, Jehan, pharmaceutical companies don't use terms that mean nothing, sativa, indica, because they think that's what consumers want to hear. They just don't label them with misnomers, and they put information into the package insert that would tell you, what a reasonable dose would be for an adult, or what a reasonable dose would be for a child, and what an adverse effect might look like if one consumed too much, and what to do if that event happened. And again, I've read a lot about how consumers needed to get more educated about cannabinoid pharmacology, and I don't think that's the case at all. I think that it would be nice if the industry did their work and put it into a package insert so that the consumer could just purchase the product, open it up, and read the salient points that are important to them. So I think, again, when you start to, say, like, some people are saying that 5 milligrams is not intoxicating, there's a lot behind that, and most of it is smoke and mirrors.
I can briefly add, I think, some of the points here taken, right, is, when we're saying, hey, industry should really be doing this, right? The beauty is, and I know we're going to talk more about the ASTM process at the end, but with this standard as an example, this is the potential opportunity. I've never seen it be any easier in the world and life to say, I have a problem I need help with. Are these products potentially intoxicating? You don't have to go it alone. You propose the standard forward as a member, which any individual in this room and watching the webinar later can do. And you might be so lucky to get 3 experts like we have on the panel today show up at your bi-weekly task group meetings to help you solve this problem and bring decades of knowledge together, because we've got over 700 individuals. So it's not industry that has to go it alone or any one company, but this is how we can work together to agree on how to solve these complex but important issues that ensure the credibility of our industry moving forward. So I think that's just an important point I wanted to make. Back to you, Pam.
Absolutely. We had a question in the chat box that I'd like to bring to the forefront. Will the standard give guidance or consideration of how to characterize in terms of impairment, intoxication, blends of products that individually are impairing, but to a different degree, with an example of Delta 8 and THCO acetate, which I know has been a big discussion point within the industry, especially from inhaled products. David, I think you have something to offer in this regard, and then we'll open it up.
Yeah, so I think my simple answer is no, but this lays the foundation, right? It establishes step one, is a cannabinoid potentially intoxicating? Once we can rule that out, if we can rule out, say, CBD, not potentially intoxicating, I'm pretty sure that's on the table under that category appropriately based on the literature and the established frameworks that we've talked about earlier today to evaluate that. At that point, we don't have to worry about route of administration, et cetera. But then once we've defined, THC, you name it, isomer is potentially intoxicating, then we're allowed to go to step two, which would be the next standard that we're ready to develop yesterday. If folks want, have thoughts about how to create that framework, it's a matter of bandwidth, I think, at this point, to get into, route of administration and dosing based on, what a accepted a general population or consumer would be, consuming and potentially seeing intoxicating effects. So that'd be step two. But we feel like we need step one here to get there. And hopefully that answers the question sufficiently. If anybody wants to add anything, please go ahead.
I think we can look at the chemical structure of the different compounds that are floating around in the industry, and we can very easily, not very easily, but we can with a good or a high degree of certainty to at least indicate whether this compound could possibly be intoxicating or not. For example, the THCO acetate, THCOA, that's an acetate of whether it's delta 9 THC or delta 8 THC. Both of them are actually on the market. There is no reason, in my opinion, why these two compounds could not be intoxicating. I mean, they are esters of intoxicating compounds. So by the fact whether they're taken orally or being taken by inhalation or by any other route, once they get into the body, they're going to convert to delta 8 or delta 9 THC, which are intoxicating. We know for sure they are. You take, for example, the hexahydrocannabinol. That's the dihydro derivatives of either delta 9 or delta 8 THC. These are already known. There is information in the literature to show that these compounds are intoxicating. So we can look at most of the tetrahydrocannabinols have the possibility of being intoxicating. Whether they are or not, for sure, this has to be determined experimentally. But just for the mere fact that they are tetrahydrocannabinols, there is a good possibility they will be intoxicated or could be intoxicating. So we can put them in that category that we can, do the next step to determine whether they are or they are not. And if they are, at what dose and how much and all the route and all of this stuff can be done later. But it's very easy to determine. Some of the natural cannabinoids that exist today, we know for sure they are not really intoxicating. For example, because not because we just think they are not intoxicating, but there is work in the literature to show that these compounds are not intoxicating. They really don't produce the same activity as delta 9 minus delta 9 tetrahydrocannabinol, trans delta hydrocannabinol. So like CBD doesn't really give those activities right away. It might interact with the CB1 receptor by some mechanism, but not really to the point where it could be intoxicating. Something like CBC, while it has some analgesic or anti-inflammatory activity, it's really not intoxicating. CBG, same thing. So some of these are already, there is some work in the literature to show they are not intoxicating. But the ones that seem to be or could be intoxicating, I think this needs to be investigated experimentally, and we determine at what level they should be administered without fear of intoxication.
So before I ask my final concluding question to all of you amazing experts, I want to make a shout out to ASTM and the ASTM process. As you heard from both David and Dr. Marcoux, it is a consensus basis. So I would encourage you to join ASTM, join the standards conversation. I like to say we learn the most from all the no's we get on the way and the road to yes. So we want to hear from you. We want to work through the issues. We know that this isn't easy, but it is critical. It is mission critical for our united cannabinoid industry. So I beg you, I implore you, I invite you to become members of ASTM and engage with all the amazing panelists that you heard today. And my final question to you all is about a vision for the future. Could you each share your vision for the future of cannabinoid science and regulation concerning intoxication? How do you envision it benefiting consumers in the industry as a whole? Kind of through this standard, but in general, if you had all of your druthers and could tell the industry how to collaborate to drive science, what would that be? Who wants to kick it off? Jayhan, you do a great job kicking it off, so I'm gonna call on you.
Sure. So I guess, my hopes for the future is that our health narratives about cannabis and cannabinoids becomes a little bit clearer. Right now, I feel like so much is at the mercy of algorithms when it comes to narratives that we forget the value of truth and how hard it is to actually find facts about cannabis and cannabinoids. I've seen so much questionable material getting lots of likes, retweets, upvotes, the strangest customer feedback I've ever read in my life. And algorithmic incentivization has really already won over so many ideals over, appropriate self-regulation. So, I understand that seeing the world through scientific lenses and as a logical place that can be explained can be a bit of a struggle. But I think we really have to start asking ourselves, how do we know what we know about cannabinoids? How are we certain we know anything about them at all, right? We have to creatively explore these questions to lead to answers. Here's a fun exercise for you to do on your own, audience. Ask yourself, what do you just know about cannabis or hemp? This product is safe, right? Or maybe it's something about your own health that you have no scientific data to support. Asking yourself these questions I think is really important. Acetaminophen, for example, no one really knows how it works to reduce pain or what dose will really work in one person. The same thing with cannabis or THC, CBD. We don't really know to describe every step of how it works to help with pain or how much is needed to reduce someone's pain with a product on the market. But yet, it is the most highly cited reason why people use these products. So I think getting down to some of these reasons of how do we know these things and why do we just know them, I think is going to be really important. So I would love to see more fundamental questions being explored in this space.
Dr. Thomas, Dr. Soli, David. So
I'm gonna jump in and follow on Jahan's comment to say that, honestly speaking, the only way we can really have a good grip on the market and and control the side effects of the cannabinoids or the cannabis products on the market is self-regulation. He put it really in the right context, self-regulation, because it is very difficult for the FDA to regulate thousands of industries. They just don't have enough agents, enough people to regulate all these industries. So self-regulation is the only way that I feel it could really work to help the public and protect the public. The honesty of the manufacturers, and they're taking responsibility for what they put on the market is the way to deal with with this industry. I don't see any other way that will be effective to really do that. So I appreciate what you said, Johan, and I know that's a very important point.
And I can join in and just say the industry tends to do a lot of thinking about formulations, and whether it's gummies, chocolates, cones, pre-filled cones, and my vision for the future is really more to what Mahmoud and Jehan just said. Everyone benefits when an industry adopts, and actually defines best practices themselves, not through consumers' experiences. And defining best practices based on did it taste good is, of an edible, is probably the least of their concern from my perspective. I'm sure other people would argue with me, but if the industry adopts best practices, follows industry guidance, ASTM in mind, and other guidance sources, and produces products that are legal, safe, and effective when used as intended, everybody benefits. It's a sure way to eliminate the black market, right? Unfortunately, we still have products that are out there making illegal claims. They're not accurately labeled. They'll put false labeling on content. They won't include all their ingredients. They'll put sativa or indica on vapes that have purified cannabinoids that have been distilled. Sativa and indica just mean nothing at that point. And they just produce confusion and distrust in the consumers. And I don't think it's, as I said earlier, it's the responsibility of the consumer to do all the homework and make sure that the product that they purchase is manufactured appropriately. They don't have the ability to do it. They don't have the knowledge about what appropriate dosing amounts and frequency should be to avoid adverse effects. And the manufacturer should help. If the industry agrees upon best practices and follows regulatory guidance, they'll generate all the requisite information and provide the scientific evidence with respect to both the benefits and the risks. And if they don't do it, they're just going to enable the black market, and they're going to put the legal market and its consumers at risk, right?
100%.
Wow. I guess I'll try to go off of those three amazing statements. I think just to build on that, for me, it's one word. I think this comes down to one word for me: certainty. And it's certainty for everybody, whether, again, you're a consumer, whether you're a regulator or your industry. Industry and businesses need certainty, right? Regulators need certainty. When there's discussions around, the gaps between a regulation, a rule, and how things are actually getting carried out, those are the gaps that create uncertainty. That creates regulatory risk for a business, that creates exposure for a business, that creates risks for consumers and what happens in the end products. And it's through the standards development process that we can get to what Dr. El Soli mentioned of, self-regulation. We can do this. We don't have to, and I don't think many folks in the industry want the regulators to come in, or the federal agencies to come in and say, this is what you shall do, right? So there's a choice. We all have a choice. You can either say, this is what I don't want, but make no effort, but that doesn't help you get towards self-regulatory structure. You have to come forward and say what you do want. We have to work through these conversations together. They have to be bound by science and guided by science. And this has worked, as you mentioned, Pam. ASTM is 125 years old. There's so many industries that have found their place in the market, in a regulated, safe marketplace, through the ASTM process and through other voluntary consensus standards processes. It's why the US Pharmacopoeia is over 200 years old and is enforced for pharmaceuticals in over 40 countries. We have the ability to make that decision and drive the agents of change together. And I'm really looking forward to that certainty. So I think that's my take on how we move forward and envision the future.
We finished right on the dot. Talk about good timing. I'm sure that all of the attendees will join me in saying a hearty thank you for sharing your expertise and your insights. I think the biggest takeaways for everyone is join us, ASTM, be an active role in the conversation, bring truth and honesty to self-regulatory environment. And I will leave everybody with the 2023 Farm Bill is the most mission critical aspect that we can effectuate this kind of change and ensure sensible regulation of the holistic cannabinoid industry.
Thank you so much.
David will provide everybody's contact information and everything in case there's follow-up questions.
Yeah, so I know we didn't get to every question that was out there, and there's a couple of items that I know folks are looking for clarity on, so I will work to get that out with our follow-up. I did just put a link in there. We updated our website so that all of our previous episodes are available in one convenient place, including the next event that will be up there soon for our October monthly series for the October event. But from there, I could not thank all of you enough, and especially you, Pam, for just being a rock star and moderating on our behalf, and for the panel bringing their expertise. Thank you all. Thank you, everybody, for attending. We will see you soon, hopefully at an ASTM meeting. Cheers, everybody.
Thank you very much. Have a great evening, and we'll see you soon, inshallah. All right.